Hello, and thank you for watching my talk on whether it takes a community to save a species and using species interactions to restore freshwater mussel species at risk. My name is Roland Eveleens, and this is part of some ongoing mussel research that I'm doing at the University of Windsor, located within the traditional territory of the Ojibwe and the Free Fires Confederacy of First Nations. I'm looking at freshwater mussels because while they're often thought of collectively, they're actually a diverse species group with many community interactions and are in significant need of conservation action. Within the lower Great Lakes, there are 41 native species of unionid mussel, of which 16 are federally listed as species at risk within Canada, due to stresses such as habitat change, invasive species, and pollution. Unionids are also disproportionately threatened globally, which is compounded by their sessile nature and need for host fish to reproduce. And so I'm investigating what community interactions need to be intact to allow successful mussel conservation and restoration, particularly what the contributions of community interactions are to populations and communities in order to better inform restoration. For the conservation of mussels, we need an understanding of which species occur together and where in order to direct restoration actions. This is especially important for mussels due to the many community interactions that they're involved in and the overlap of species habitats leading to many species being found together at range scales. Firstly, we need to understand which species occur together after environmental and biotic filters shape the potential species pool into the realised local community assembly. For example, the occurrence of suitable habitat conditions or host fish. And then we also need to understand the trophic interactions that occur between mussels and other species groups, including other benthic invertebrates as well as fish. Now, degraded communities are likely missing species and so may lack the interactions needed to support mussels. Therefore, restoration seeks to restore these connections to recreate self-supporting ecosystems. And that's what I'm going to focus on here, as we need to understand both the trophic and life cycle interactions taking place. To utilize such interactions for individual species, we also need to understand how species traits play a role here. Host fish display known traits that affect mussels, such as host fish specificity for the host fish mussel relationship, as well as the dispersal distances of host fish. Mussels also have diverse assemblies and traits, such as differences in morphology, size, growth rate, and habitat preferences, as well as displaying different reproductive strategies, such as equilibrium, opportunistic, or periodic strategies. And so understanding the traits of persistent species can inform the conservation of other species. The relationship of benthic invertebrate traits to mussels is also not particularly well understood, beyond the potential for mussel ecosystem functions to alter nearby invertebrate communities. However, invertebrates are key components of aquatic food webs and may display shifts in resource use and functional feeding groups in response to similar stresses that affect mussels. And so I'm particularly interested in the interspecies interaction between mussels and the interactions between mussels and benthic invertebrates to understand whether these need to be included into conservation and restoration. For that, I'm looking at associations between mussel species, patterns of mussel community assembly over environmental and habitat size gradients, and the link to benthic macroinvertebrate communities, and specifically differences between sub-watersheds and between tributaries and main stems. I'll refer back to these three icons on the left of my slide for each of my aims as I work through my results. And for geographic context, the work that I'm presenting here today was done in the Sydenham River, which is within the North American Great Lakes and flows into Lake St. Clair. It is a hotspot of mussel diversity within Canada and features at-risk mussel species such as the kidney shell, rayed bean, snuff box, round pigtoe, and northern riffle shell, to name a few. Over the 2020 summer, we surveyed 15 sites across the Sydenham River following a hybrid protocol to increase the potential to detect species, which involved a 4.5 person-hour time search at every site, and if we found an abundance of mussels or species at risk, we also conducted a quadrat survey. Benthic invertebrates were sampled following a standard provincial Ontario benthic biomonitoring network protocol, while environmental data was also collected following standard provincial methods in order to produce data sets and analyses that were more widely applicable. In terms of the environmental conditions, this spanned from sort of large, slow and silty sites on the North Branch watershed to smaller and still slow and silty sites in the Black Creek watershed. And then the East Branch subwatershed had larger and more flowing sites with coarser substrate, although it also had sort of smaller and siltier tributaries, which with the locations of these sites, so relatively indicated by the letters on the inset map. The land use within this watershed is predominantly agricultural, so sediments and nutrient runoffs are a significant water quality concern, and there are also strong differences in substrate and riparian conditions across sites. As I was lacking the environmental data to robustly define a continuous gradient of environmental impact, I grouped sites into categories based on sub-watersheds and watershed position, comparing river main stem sites with tributaries that had a stream water of 4 or less. Now, these groupings had significantly different environmental conditions when tested using a principal component analysis. And we also saw a difference in species richness across these different groups of watershed position, with greater spread of species richness in main stem sites compared to tributaries, and also across sub-watersheds, as East Branch sites were the most diverse, followed by the North Branch and Black Creek. Here, asterisks denote significant differences between groups when tested using an analysis of variance. Now, the same pattern of differences in species richness was present when we looked at just species of interest to restoration, including species at risk and also species listed as special concern, which were only found in the main stem and were more diverse in the East Branch. And this held true when we just looked at species at risk by themselves, although the sample size became too small to test the significance of these differences between groups. And so therefore, this tells us that there's a significant difference in the diversity of main stem sites compared to tributaries and between sub-watersheds. When we looked at the overall community composition using ordination, where the abundance of every species at each site was combined into two-dimensional space, so that each point on this plot represents the mussel community present at a site. And so the closer together points are, the more similar the mussel communities found at those sites are. We found that when tested using permutational analysis of variance, there was a significant difference across sub-watersheds, which is shown by the different colors on this plot, and also between tributaries and main stem, shown by the different shaped symbols. Now, when we looked at which communities had species at risk or species listed as special concern present, we also found a significant difference, with the presence of species at risk or species listed as special concern only found at our larger, more diverse sites. So that showed that there was a significant difference in the community composition of mussel communities where the species at risk were present and where they weren't. So this difference in assembly led us to thinking about environmental drivers and the involvement of community interactions in these, and particularly investigating species co-occurrences more closely. When we looked at the relationship between mussel communities and species at risk occurrence, overall mussel species richness was higher at sites where species at risk were present, and that stayed true for the presence of all individual species at risk species, and species at risk richness was highly correlated with total species at risk, mussel species richness. However, when we looked at the relationship between benthic macroinvertebrates and mussel diversities for the sites sampled just in the East Branch, there wasn't an ecologically meaningful relationship between benthic invertebrate family richness and mussel species richness. While both family richness and waterway position had significant effects, waterway position mediated the effect of family richness because it explains a much greater proportion of the variation. And when we looked at the effect of family richness alone using a partial regression plot, we could see that the slope was near zero. This non-meaningful pattern remained for other benthic macroinvertebrate diversity metrics, including the Hilsenhoff biotic index of organic pollution, and both the richness and relative abundance of more sensitive mayfly, stonefly, and caddisfly taxa. But certain taxa were indicative of mussel species at risk presence or absence, as identified by using an indicator species analysis, which identified taxa present at the majority of sites within a single group. And so 4 invertebrate families representing multiple orders and functional feeding groups indicated the occurrence of species at risk, while the presence of Corexidae indicated the absence of mussel species at risk. And so from that, while there was no difference in the benthic invertebrates, no relationship between the benthic invertebrates and the mussel communities for the subwatershed we investigated, we did observe clear differences in mussel species assemblies across subwatersheds and with watershed position. So while we already know that environmental conditions and host fish presence play a role here, the occurrence of species at risk at only some sites with diverse communities suggests that the biotic interactions are or were present to support them. And I'm continuing to unpack any patterns between species as well as between mussels and benthic invertebrates. In terms of our traits framework, we know that fish have known traits affecting mussels, with species at risk relying on specialist host fish with small dispersal distances. And so fish matter for restoration. And while mussel interspecies interactions need considering, as there are clear patterns in assemblies, but not so much in reproductive strategy, we're still asking if co-occurring species are reproducing and sustaining those diverse assemblies at the sites we found them, and if there are similarities in other species traits. And it's still unclear how much invertebrate traits relate to mussel species and assemblies, and so their role within restoration, as the mussel-invertebrate relationship appears dependent on environmental context. However, there's still key indicator taxa connected to the occurrence of species at risk, which suggests they could indicate suitable habitat. And so while I'm unable to clearly identify a key suite of traits to prioritize for conservation, this does start addressing some of the mismatches between research and practice that were evident from a separate systematic review of mussel restoration tools that's currently in review. It shows that clear differences in sub-watershed and clear differences in mussel communities across sub-watershed and watershed position matter for mussel community composition and can direct the location of conservation and restoration actions. And the occurrence of species at risk in diverse assemblies suggests that habitat quality may be more important than specific environmental conditions in order to support the co-occurrence of multiple species. And there is potential for faster responding indicator species to be of use for identifying these high quality sites. And there also poses whether there is a need to translocate multi-species assemblies together in order to preserve positive interactions. And so I'm looking forward to continuing to address these questions as I continue my analysis. And with that, I'd like to finish and thank you for listening. Thank my additional committee members, Dalen Wilno and Forever Picture, the organisations that have funded and assisted my work, all these lovely folk for continuing to help me with my project, and the landowners that allowed access to my survey. I'm happy to answer any questions, so please feel free to get in touch through the conference platform, Twitter, or email. Thank you.